So, Bob Lefkowitz, Brian Kobilka, welcome to Stockholm. And indeed a snowy, wintry, beautiful Stockholm. It couldn't look more inviting, but very different from North Carolina and from the Bay Area this time of year.
Indeed. We only get snow maybe once every 3 or 4 years, so it's been kind of a treat.
And so you're here for Nobel Week, the culmination of 2 months of fairly frenzied activity, I imagine, since the calls came. How have those last 2 months been? How have you found it?
A bit overwhelming. It would have been a busy 2 months had this not happened, but it's been an extraordinarily busy time, yes.
I'm sure. And how about you?
I had been warned as soon as this happened. I spoke with several colleagues, Harold Varmus and Joe Goldstein, old friends and colleagues, and they warned me about just how intense it was. But I must say it was, like as Brian said, I found it a bit overwhelming right up to our arrival here. So it's something that nothing, I think, prepares you for.
No, I suppose not. And you had the added onus of being the first Nobel laureate from Duke University.
Yes, indeed. And so this caused quite a bit of ruckus, that it was a first. And they likened it to when we won our first NCAA basketball championship under the very famous Coach K, Coach Krzyzewski. And I think one of the most exciting things that happened to me during this period of time was that I was honored at Cameron Indoor Stadium, which is the basketball stadium, by Coach K and the basketball team that presented me with a jersey with a number 1 on it and my name. Which seemed to impress my children, perhaps even a bit more than the Nobel Prize
I'm sure,
itself.
Yes. It's nice that intellectual pursuits and athletic pursuits have reached a level. Yes,
Indeed.
That's good. And it's hard to count Nobel laureates from institutions because there is always, one never really knows whether people are associated. But I've heard Stanford themselves saying that you're number 27. So
That's probably right. Yes, it's not quite as big of a deal there, but
I'm sure it's still a very big deal. One thing that often is said is whether it gets you a parking space on campus.
I'm afraid not.
Not yet. Maybe you need 2 prizes for that.
How about your own bathroom? Do you get that?
No.
Me neither.
Anyway, you've been awarded for your work in the field of Gproteincoupled receptors, GPCRs. And these are the gateways to the cell together with ion channels. And it's commonly said that about 50% of medicines act through Gproteincoupled receptors. And it's hard to imagine really that 40 years ago, when you started working in the field, that the concept of the receptor itself was still not all that widely accepted. There was still some doubt as to whether they existed.
Yes, there was a great deal of skepticism about this. And in fact, in my Nobel lecture on Saturday, I'm going to review some of this. I mean, frankly, I could give the whole lecture on that topic, but I won't. But remarkably, the skepticism was shared even by some of the giants of the pharmacology field, such as Raymond Dahlquist. This was a very famous pharmacologist, American pharmacologist, who won the very distinguished Lasker Prize for first putting forth the idea that there could be 2 types of receptor for adrenaline, which he called alpha and beta receptors. That was in 1948. And that was based on some classical pharmacological work that he had done. But as late as 1973, which is when I was just sort of beginning some of this work, he published an article basically saying that it was essentially arrogant of people to even think that these things really existed. For him, they were just sort of an idea, a way of organizing thinking. But he didn't really, and I think this was the kind of, there wasn't really a concept that these were specific molecules, if you will. They were just sort of viewed as some very, I don't know what word to use, not diffuse, but unclear, nebulous pattern of forces on the surface of cells that could somehow allow them to interact.
Well, that's interesting because the first half of the 20th century was really the golden age of neurotransmitter research.
Yes.
And this slew of neurotransmitters were discovered and their functions were to some extent understood, and yet nobody knew how they operated and
This is correct. And in fact, the earliest idea about receptors came from a British pharmacologist named J.N. Langley. And more than 100 years ago, he wrote about the idea that there must be something that we now know as a receptor. But interestingly, 40 years later, his student, Henry Dale, who, talk about neurotransmitters, he won the Nobel Prize, I'm not sure what year, for his work on cholinergic neurotransmission. And in the mid-40s, he wrote an article mocking his mentor, Langley, for saying that there was such a thing as receptors. He says, it doesn't really teach us anything to say they're receptors. He says, of course, there's something about cells that allows them to interact with molecules, but giving it a name just doesn't really make any sense.
So why did you choose that field for your own research when you started research?
I had been at the NIH for a fellowship in 1968 to 1970. And this was the golden age of second messenger signaling. Earl Sutherland had recently won the Nobel Prize for his discovery of cyclic AMP, which is a molecule generated on the inner surface of the cell membrane when what we now know are GPCRs are activated. And my mentors there, Jesse Roth and Ira Paston, thought it might be possible to label the receptors, because they felt they existed. They were what would be called molecular endocrinologists. And they thought it would be possible to label them with radioactively labeled materials. And so I was assigned the project of trying to label a receptor for ACTH, which stands for adrenocorticotropic hormone, which works through a Gproteincoupled receptor. And I spent my 2 years there radioactively labeling ACTH and showing that I could bind it to some sites on the plasma membrane of a tumor, which was responsive to ACTH. So it was an ACTH responsive cancer that I passaged in nude mice. And so we were successful in doing that. The work didn't really go any further than just developing these radioligands, but the idea caught fire in my imagination.
Gave you an inkling that we could
do this, right? And then when I went further in my career, I was a cardiology fellow a couple of years later, and I really wanted to pick up this theme, but I wanted something more cardiovascularly related, and the adrenergic receptors seemed to fit the bill more so than ACTH. So that's why I turned to the adrenergic receptors in the early 70s.
Okay. And so I'd just like to pause there, because one thing that defines you both, you were both cardiologists.
Mm-hmm. I had the intention of becoming a cardiologist.
Okay.
What are you talking about? You did the fellowship.
Yeah, so then I started out in Bobsled, and that was pretty much, with the exception of the intensive care unit, the only thing I really did to complete my cardiology fellowship, which I didn't complete.
But you started out with the intention of becoming a doctor, a medical doctor, and then you transitioned to research. So I was going to ask you both what sort of flipped you away from medicine and to the bench.
Well, I think I had the intention of, at the time I was in training in internal medicine in St. Louis, of being an academic physician, which meant that I would continue seeing patients and I would find some line of research. And at the time, I was really very interested in intensive care medicine. And I believe there weren't intensive care medicine fellowships at that time. It was either cardiology or pulmonology, and so I decided to go into cardiology. And I had learned that I had also a real interest in research, although I can't at the time say I was really interested in adrenergic receptors. I was interested in the concept of using adrenaline and other compounds like that in the intensive care unit. And Duke had a fantastic research program for cardiology fellows. You could go to Duke, you could spend at least a year and a half doing research, and you could do it up front, which was really attractive to me after spending 3 years taking care of patients and really wanting to try out research. So I don't know how many places I applied to, but I was very happy to be accepted into Duke, and very happy that Bob chose to let me in his lab.
We'll come to your lab environment soon. But so when was the last time you saw a patient?
Well, I continued to see patients while I was a fellow, primarily moonlighting on weekends. And I'm not sure, I think I can say this now since the statute of limitations is up. But when I moved to Stanford, my wife Dong Sun started medical school. So our children were old enough to go to school. She started in medical school in Stanford. So we had a very large mortgage and tuition, and I continued to moonlight for quite a few years in emergency rooms.
Okay, so maybe you're still moonlighting. Maybe we shouldn't talk about it. And how about you, Bob? You had wanted to be a doctor.
I very much wanted to be a physician and had harbored that goal, I would say, from the time I was 8 or 10 years old. Like Brian, I learned last week a very similar experience. I was inspired by my family physician who made house calls, and I decided that's it. That's what I want to do. And I thought about nothing else in terms of a goal right through grade school and high school and college. I would say that Brian, from what I know of him, actually showed an interest in research earlier than I, because as I understand, Brian had a mentored experience in college, and I think he even won an award for his research thesis at Yale, if I'm not mistaken. I had absolutely no research experience until I went to the NIH in 1968. In fact, in medical school, we had a couple of opportunities to do 2-month electives in research. I never took one. I had no interest in it. I wanted only the clinical electives, the so-called sub-internships. But in the late 60s, the Vietnam War was raging, and many of us were opposed to that war on multiple grounds. And so there was a draft, and physicians were drafted, and you had to go in after 2 years of training, which many of us didn't want to do. So there were a few options that were very competitive to get, that would get you out of going to Vietnam. One was to join the United States Public Health Service as a commissioned officer and be assigned to the NIH for 2 years. Very, very competitive to get such positions, but I had a strong academic record and I was able to get that. And it seemed like a good thing to do because, like Brian, I pictured myself someday as an academic physician, and I vaguely had it in my head that academic physicians did some research. So I figured, well, I'd get my research papers and I'd go. And that's how I got started in research. Now, there's a very interesting and timely piece in, I guess, last week's issue of Science by Mike Brown and Joe Goldstein, in which they review basically the history of that era. And remarkably, it turns out, between 1964 and 1972, I think is the slice they took, I become the 9th Nobel laureate to have trained. And I actually went through the list. 6 of us actually came between 67 and 70. And when I say came, I don't mean like there were 100 of us each year. In a year, there might have been 8 or 10 or 12. So it was a remarkably high success rate.
And they go on in that article to regret the loss of that kind of training environment.
Exactly. I mean, it was a remarkable environment. They tried to dissect what it was about it that made it so remarkable. And I think they do a pretty good job, but, you know, there's some things that can't be fully understood. I mean, obviously, they got the best talent coming in because it was so competitive to get the positions. And there were some wonderful mentors there. But I don't know, there may have been something else that's harder to define.
Anyway, avoiding the draft, in fact, led you to a research career. Somehow.
Exactly right.
Exactly.
It's not what I had imagined at all. And I certainly didn't have a smooth start. I mean, things went very poorly at first.
Okay. And when did you give up on patients?
Well, I actually made teaching rounds for 30 years at Duke. Okay. I would do 1 rotation a year. It was 6 weeks. And then from age 30 to age 60. And after my 60th birthday, for a variety of reasons, I hung up my stethoscope.
So at Duke, you pursued this incredibly difficult task of purifying the beta-adrenergic receptor. It took 15 years to purify and clone. That's quite an undertaking. Did you have, as you were on that path, the goal of that purification so that you were always looking that far ahead?
definitely that was a goal for many, many years. And something interesting that, at least it's my view of things now, and maybe it's revisionist history, but when I look back on that period, my sense is that it almost never occurred to me we would fail. Okay, I mean, I always believed we would be successful, which when I look back on it now, it was kind of crazy. But that's my sense of it now, that I always assumed it would all work. I didn't know when or how long it would take, but sooner or later we'd get there. I mean, it never occurred to me that maybe it wouldn't work at all and it would be a total dead end.
Sure. But along the way, you have to be generating enough material that keeps it all moving.
Moving, yeah, right, exactly.
So, yeah.
And I think Brian had, a similar experience these last 15 years with his crystallography. Although I don't know, maybe you did entertain the idea that it wouldn't work at all. Or did you always think, someday I'll get there?
I think I always thought someday I would get there.
Yeah. I think that kind of optimism, what did you call it once?
Henrik, a colleague of mine from Bobsled, calls it irrational optimism.
Irrational optimism. Yeah, I mean, sometimes that's, I think it's a good leadership quality because I think it's sort of infectious. And I think in terms of the troops, the folks in the lab, I mean, if they think the boss has any doubt we're going to get there, I mean, that doesn't foster the kind of attitude that you want.
No. Okay, well, let's talk about the lab a bit, because you've had an immensely productive lab, both in the sense of papers and discoveries, but also in the sense of people. You've had, what, more than 200 people?
Yes.
So you must have got a research environment going which people want to join. What's the secret of it?
That's a very good question. I think there probably are a number of elements. One, of course, is the old business of success breeds success. I mean, if a program is successful, then people want to come because there's high visibility research going on there. As to why we were so successful, I mean, there's a lot of mystical stuff. I mean, like, I'm a great judge of talent. That's how Brian came to my laboratory. I say that and we both laugh because when he came to interview, as I recall, I was out of town, right?
There, yeah.
I wasn't there. And I think he may have even made a second visit and I was also not there. But in any case, I wasn't there. So obviously I'm a good judge of talent, and I took him anyway, sight unseen. But I think one of the things about working in a lab is to have good esprit de corps, which can't be 100%. There are always going to be some oddballs or some oil and water mixtures of people just don't get along. But I think in general we had good chemistry of people in the laboratory. And I think I always brought to the endeavor a real sense of enthusiasm for what we're doing. Enthusiasm is something that you can't fake. You either are, you can't fool people. And I remember at my 60th birthday party that Brian was at, and many of the fellows came to, they were giving various talks about what they remembered. And I remember one postdoc from that era, a wonderful guy named Rick Serione, who's now a professor as a named chair in chemistry, actually, at Cornell, was saying that one of the things that kept him going is that he always sensed from me that his project, which was the reconstitution of the receptor after it was purified, was the most important project in the lab. And he liked that. Until one day he talked to some other guy, and he said, well, no, I mean, I felt my project was the most important. Then it turned out everybody thought their project was the most, and I guess somehow I transmitted that because probably I believed it, including Brian's, which really was the most important project in the lab.
So with Bob sitting here, maybe it's not the right place to ask you, or maybe it is the right place. Does that tie in with your experience of Bob's lab?
I would say yes, yeah. You really had, he was always very enthusiastic, encouraging, and you felt that you could try anything. I don't actually remember there being any kind of limit on even what we could spend to try something that was completely, sometimes crazy. Yeah.
I think one of the things that I taught Brian was how to go into debt in the laboratory, and he's been very good at that in recent years, which I'm sure he could tell you about. But the folks in the lab probably didn't even know it, but on several occasions when things really got hot and we were making progress on a project, I would just overspend. I mean, I just wouldn't, what he says is true, I would not care about the budget. We would just do what we needed to do. And I was called on the carpet on several occasions, both by the chairman of medicine, named Jim Weingarten, and also by the head of the Howard Hughes Medical Institute. On several, the administrator of the Hughes Institute then was a guy named Kenny Wright, and he was a good old Southern boy. And I remember on 2 occasions they sent him down specifically, as he put it, to slap me on the wrist and take me out behind the woodshed because I had totally overspent my budget, and he was there to sort of rein things in. On the other hand, we were doing good work, and et cetera, so he just kind of gave me a wink, but you know, he brought the message anyway.
Mm-hmm. Mm-hmm. So I know that, that you have a very large number of people coming from all over the world to help you celebrate here. You've got this get-together of 50 plus people on Sunday.
Yeah, at the moment it's up to 55 with a few family members.
So I suppose that speaks to the social aspect of all this, that it's not just productive and kind of supportive, but you're also becoming friends and lasting relationships. And I'd like to talk a little bit about that.
Yes, well, and I think Brian will have a lot to say about this as well. I think both of us have tended to view our laboratories as a second family. I mean, you really get close to these people, and they really are. You know, as a mentor, they're like your kids. And like your kids, when they leave the nest, doesn't mean you're finished with them. You know, it goes on for a lifetime. And then I think some of the relationships amongst the trainees that they make within the laboratory, people who are in the lab at the same time, I think become colleagues and friends for life. And it's a very, very special bond. I think, you know, in my own laboratory, probably if there was a peak period when I think things were rolling, it was really the 80s and into the early 90s, which encompasses the period when Brian is there. And many of the people who were in my lab at that time are acknowledged leaders in our field and are good friends and colleagues of Brian. And I think it speaks to the way they feel, not just about me, but about Brian, that they're all coming here, you know, at their own expense and unable to get into the ceremonies, but just to sort of be here with us, I think speaks to that family aspect of things.
Because in a way that's at odds with how science might be often popularly can't say that word, perceived by the public. Right. That you've got the image of the lone scientist coming up with ideas and pursuing their goals, and indeed that's in some ways supported by the Nobel Prize, which selects individuals for the prize. And yet, of course, it is a very social affair. And in your case, even a family affair, because you and your wife work together in the lab.
That's true. It's hard, you know, there's no conscious decision that it's going to be that way, but it seems that, that at least in part of my success is finding people that both I enjoy working with and are willing to engage in very challenging projects. And my wife in particular, I would say particularly during the past decade, where some of the projects in the lab were too risky for our graduate students or postdoctoral fellows, she would be rolling up her sleeves next to me and working on these projects and also helping out fellows and graduate students with really difficult projects to make it at least more feasible for them to engage in these high-risk projects.
So when you pick people to join your lab, what do you look for? People you're going to get on with, first and foremost?
It's very important that not only I get along with them, that they get along with other people in the lab. It's really important to have chemistry in the lab. And I would say that the lab has been most successful when people work together on, again, very challenging projects, sharing ideas, sharing reagents, really teaming on projects. And yeah, I think that's really, really key to having a productive and enjoyable experience.
I think something that's very important both to Brian and myself is the mentoring. of these, the young people. And people often ask me, because I have been successful in training so many people, you know, what are the keys to mentorship? But it's like everything else. There's not one right way to do it, and just like there's not one right way to do science. I mean, I think many people would consider both Brian and myself very good mentors, but our personalities couldn't be more different. So obviously we can't possibly mentor people in the same way, and yet we both seem to wind up doing a fairly good job of it. So I think a lot of it is just a matter of, are you willing to invest yourself in people and really care about them and care about their careers and success and challenge them. I mean, to put things in front of them which they really have to reach to get to. And I think, you know, if you can give somebody the experience of working right at their potential, okay, in other words, really doing something which tests them and extends their limits, and let them feel what it's like to succeed at that level, then you've done it. Because then they know what it feels like, they know what they're capable of, and they're willing to challenge themselves in the future.
You have to know them pretty well to know what to challenge them with.
Exactly. I think getting to know, or at least for me personally, getting to know people and sort of getting in their head and really, because it's different. You challenge different people in different ways. It takes different kinds of things. And the whole issue of how much to direct people, I've known it's a delicate balance. If you can direct them too much, and they may be successful in the short term, but they never gain the confidence that they can really do it by themselves. Or you can direct them too little, and they just kind of drift and become unfocused, etc. So the question is to provide enough direction, but not too much direction. And of course, there's nobody who can tell you what that is. You just sort of have to figure that out individually.
But it must be a lovely feeling when you see it work and you see the person fly.
Absolutely, absolutely. And I'm aware of some very, very distinguished scientists who, in the course of long careers, have not trained that many successful people. And you say, why is that? And at least in the couple of cases I'm thinking about, I think I know the answer. And I think it's because they so closely directed their trainees while they were there that they didn't really have a chance to develop their own independent thinking and confidence to really do things on their own. So
one other aspect of this, and then we'll move on, but by generating 200 plus people from the lab, I suppose you're also generating, this is a bit of a nasty thing to say, but 200 competitors. You're seeding a lot of people who are interested in the same stuff.
Well, absolutely. In a sense, it's true. So then it becomes a matter of competition is in the eye of the beholder. So, I mean, Brian, when he left my laboratory, continued along doing things that I might have worked on. Sometimes I did work on. But I guess it's sort of like with your kids. I mean, I can't imagine ever feeling competitive with one of my kids, even if they were doing exactly what I was doing. It's just, you know, it's sort of like, it just doesn't work that way. So again, I think that's the familial aspect to it. Yeah, I mean, how could any of them compete with me? They're like my kids.
Yeah. And you've been in a pretty competitive place for the last 15 years going for this crystal structure, because there were other big groups heading down the same road. But in this case, there's not the familial bond. They're just other groups out there. So.
Yeah, and I'd have to say that you know, there's been even among some of my competitors been a great deal of collegiality and collaboration as well. I mean, I would say particularly with Gebhard Schertler, who was one of the earlier structural people in GPCRs. In fact, he generated the first structure. It was what we call a two-dimensional crystal, and it was a lower resolution structure, but it was really our first view of what a GPCR looked like. And he's always been a great colleague. He in fact took us to the beamline when we got our first crystals, which weren't good enough to get a structure, but they were small and too small to actually study on a conventional synchrotron. And Gebhard was really interested in developing what we now call microfocus or microbeam crystallography. And he had worked with scientists at the ESRF on the first really high-quality micro-focused beam, sometimes called mini beams. And he took what were pretty lousy crystals, and brought me to his synchrotron time. We went to ESRF. Tungsan joined me on the trip, and so he really taught me how to start collecting data from very small crystals. And we've been friends and competitors in some respects. Yeah.
Brian makes a very good point. I found in my career that competition per se doesn't necessarily, I'm talking about outside the family now, competition per se doesn't necessarily mean, difficulties. I've had direct competitors who I've had lovely personal relationships with, and I have had others which were not so pleasant. So it's not the competition per se, it's more a matter of the personalities, I think.
I suppose that in some ways it's there to keep you at the top of your game. You have to.
Yes.
Yeah.
Well, there's no doubt that competition does drive a lot of science. I mean, I think all of us are inherently somewhat competitive.
So if we just return to the lab, and you came into the lab and were involved in the latter stages of this beta-adrenergic purification and cloning experiment, where the structure of the beta-adrenergic receptor was revealed. And that acted, in a phrase that sort of seems very appropriate, as the Rosetta Stone, unleashing lots of other G-protein-coupled receptor structures and revealing that there was this superfamily out there. I've often heard people say that in retrospect, of course there was a superfamily, it was obvious, but was it obvious as you went into it?
Not in the least. When we were cloning, when we had first been successful in getting what we knew to be valid clones for the receptor, it's not like today. It's amazing to think back. How many nucleotides were there in the open reading frame? A couple of thousand.
Yeah, 1,500.
Yeah. And how long did it take between us and Merck to sequence that piece of DNA?
I don't remember.
It wasn't a day or two, was it?
No, it wasn't a day or two. No.
Yeah, it was probably a matter of weeks.
Probably between 100 and 200 base pairs at a gel. And, yeah.
Yeah, I mean, so while we were sequencing it, word began to get around on campus that I don't know if you know this story. So word got around on campus. We had it, okay. And I don't remember who, but somebody, so we had part of the sequence. I don't know how many base pairs. And we knew we had the right thing because we already could see some of the 5 peptides that we knew were in there from our sequencing of pure protein. And a colleague said, "What does it look like?" And I remember distinctly saying to him, "It doesn't look like anything." I said, "Why should it look like anything? This is the first one." Okay, so in other words, that just shows you the extent to which we were not expecting what we eventually found, which was the homology with rhodopsin. This, even though the functional analogies between rhodopsin and transducin and the cyclic G-phosphodiesterases with the beta receptor, the G protein, those functional analogies were clear. And I think by then it was probably clear that transducin and GS were members of a family. Nonetheless, nobody was expecting the beta receptor and rhodopsin to look alike. And it was only later, after the full sequence emerged, that these homologies became clear. But in the early going, I thought we were describing the very first, that it was sui generis.
Mm-hmm, mm-hmm, mm-hmm. And was it with great pleasure that you realized that you were suddenly opening up this superfamily, or was it in fact a sense of, oh, other people kind of knew this already, and it was that that other people could have twigged this?
Well, I mean, shortly after we reported our clone, about 4 months later, Elliot Ross in Dallas reported on another, the beta-1 adrenergic receptor from a turkey, and it looked very similar. And then Shosaka Numa, a little bit later, reported on 2 muscarinic receptors. So very quickly, within a year, or less, we had those. And then Brian cloned the alpha-2 adrenergic receptor, again, within about a year, based on protein sequence that we had. And so very quickly it was filling out that there was this family. I think in the moment we cloned the beta-2, I think it was a eureka moment, but I don't think any of us, and we said right in our first paper, maybe they're all going to look like this, but I don't think any of us really conceived just how large this and diverse this family was going to be. And certainly, of course, the olfactory receptors, which came a full 5 years later, like doubled the number of receptors right on the spot. So I think, it's like all discoveries. I think it's, well, not all, but it's like many discoveries. You don't fully appreciate the significance in the moment. It becomes clearer and clearer and clearer over the years.
And so your lab continued to discover things about the structure and function of GPCRs en masse, so to speak. There were lots and lots of discoveries pouring out. Meanwhile, you then moved away and went to Stanford and began this quest. And what you eventually produced was the high-resolution structure of a GPCR with ligand and G protein attached. In other words, the whole kit and caboodle. And I remember in 2004 you commented in an article that we asked you to write that you thought you might be a year or two away from the structure. And I guess there were ever higher resolution structures, but it seemed to take longer than you thought it might. Must have been difficult going.
Oh, yes, it was difficult. In 2004, I think we were starting to get crystals, so I felt that, and I'm not sure I said that we would get it. I said somebody would get it, and there were of course a lot of rumors.
Yeah, Brian, you were telling me, I remember that
yes.
There was a rumor that so-and-so had it and so-and-so had it, and you were quite nervous about it.
There are some really detailed rumors, which I don't know how they ever originated, but so there was, I knew that we had succeeded, and other individuals were telling me that they had crystals, and so I think it was a logical assumption that it would happen sooner. Obviously, it took much longer than I thought.
It was a good sign at that point, though, that you were thinking about it harder than other people, because, or at least as hard as anybody else was, because the question I think we asked was, when will the structure come, and what needs to be done in order to get there? And we asked 20 people to answer that, and they wrote about this much, and you wrote about this much. So you, you obviously had a lot of thoughts about it.
Well, I had been working on this project for a long time, and not just crystallography, but trying to understand the protein using other approaches, particularly biochemical and biophysical approaches. And so while these were much lower resolution, particularly fluorescence spectroscopy, really taught us a lot about the protein, how it behaved, and I think in many respects that experience with these different approaches taught us a lot about what we probably needed to do to overcome the obstacles we were having with getting to high-quality crystals, and ultimately informed us about the different approaches that ultimately succeeded.
But one other thing that struck me at the time, this was an article that was written for one of the Nature journals that you said this in, was that you were very giving with your suggestions. You seem to be revealing quite a lot about the way you were thinking and the answers you gave. And it speaks to that idea of being a collaborative person rather than a secretive person.
I hope that's true. Yes. Of course, I wanted to be the first, and I probably wasn't completely revealing of course not. But
it's interesting when I think back at the situation Brian found himself in, I guess around 06, 07, when it was clear the field was converging on a crystal structure. And there were several players, and nobody knew who would get there first. But to go back in time to 1985, 1986, as a very young fellow, he was actually put in a very similar position because we were in a direct competition with probably several labs, but we knew about one. And early on, they were kind of ahead of us. But then, because of a bunch of circumstances, we leapfrogged the whole thing. Things happened very fast for us when they finally happened. But it was a similar kind of deal where there was a lot of pressure, intense competition, who's going to get there first. So he basically had that experience once before at the very beginning of his career. And now here he was, whatever it was, 25 years later, in the same position. And he and I talked. I tried to advise him as best I could. I wasn't involved in the science, of course, but just as sort of an older head. It's like I always like to tell people that the key to Brian's success is that whenever he has important decisions to make, he discusses them with me and then does the opposite. And this has led to a great deal of success on his part.
Everybody needs a sounding board.
Right.
Well, you obviously have an appetite for these high-pressure situations. Right. We don't have very long left, but I would like to just touch very briefly on the medical aspects. As I said at the beginning, that about 50% of medicines are thought to act through GPCRs, but there are a lot of off-target effects, there are a lot of side effects. Would you like to say how the work that you do has impacted or will impact the development of medicines?
Yes, so I think that in particular the recent work that both Brian and I have been doing, totally different kinds of work, each in their own way has implications for potentially developing more specific medications, by which we mean medications which have less side effects. So if you look back, I think, over the history of pharmacology, at least during the 40 years that I've been involved, there have been several advances that have helped us to, by us I mean others, I mean the drug industry, to tailor GPCR targeted drugs with less side effects. The first development was the elucidation of previously unknown receptor subtypes. And the cloning revolution changed everything. I mean, when I started my career, I think there were 3 adrenergic receptors, and then early on, I guess right around the time in the early mid-70s, a 4th was discovered. By the time we got done with our cloning, we had 8, and then a 9th was added. Okay, there was 1 dopamine receptor, it went to 5. There was 1 muscarinic, it went to 5. There were 1 or 2 or 3, I forget, serotonin receptors, it went to 15. Okay, and with the advent of these greater and greater receptor subtypes, you could get more and more specific actions because you didn't have to get the off-target effects from a different subtype. But now I think there may be even other ways in which we can get greater specificity. One of the things I've worked on for the past decade has been the fact that in addition to signaling to the inside of cells through G proteins, the receptors can also signal through a different molecule called beta-arrestin. This is a molecule that we discovered about a little more than 20 years ago as a key part of the mechanism which turns the receptors off. Well, it turns them off with respect to G protein signaling, but at the same time, we've only come to appreciate recently, serves as an alternate signaling mechanism. Now it turns out that you can design drugs which will activate signaling either through the G protein or the berrestin, through the very same receptor. So for some drugs, it turns out that the desired effect through that drug actually is being mediated through G proteins. But some of the side effects are being mediated through beta-arrestin signaling, or vice versa. So if you had a drug which can target one or the other mechanism, you may be able to get fewer side effects. And in fact, there are several examples of such compounds which are now in early phase clinical trials. So that would be an example of how my work in recent years may have implications for getting even more specific drugs with less side effects than we've had.
Thank you. And as for the crystal structure?
Well, I think there are 2 ways that we might facilitate the development of more selective drugs. One is, as we learn more and more about the binding pockets, particularly how related receptors are so similar to each other in terms of the amino acids that form the binding pocket, we're discovering that just outside of the binding pocket, there's greater diversity that could be sampled, and some drugs do sample this. For example, it's a structure that we recently obtained that hasn't been published yet is a drug called salmeterol, which is used to treat asthma. It's extraordinarily selective for the beta-2 over beta-1, and it's partly because it extends out of what we call the orthosteric pocket into more of the extracellular surface and samples differences between beta-1 and beta-2. So I think that's an opportunity for, these structures will give us an opportunity to actually design drugs that might be more selective. The other is to take advantage of unconventional binding pockets. Well, first of all, there are also allosteric binding pockets that are, for example, the muscarinic receptors are well known. But there may be other druggable surfaces on the receptor, including surfaces on the inside of the receptor that we might explore that will have greater structural differences from even closely related receptors, say for the beta-1 and beta-2. So those are a couple of opportunities to develop more selective drugs.
So for the first time, it's really possible to use the structure to do structure-aided drug design.
Yes, and I think a colleague of ours, Brian K, has really been at the forefront of using computational approaches and shown, as he likes to say, that GPCRs have really great binding pockets for in silico screening.
Okay. Thank you. Okay, and just to wind up, something that happened on that day in October when you got the call was that you were both converted into chemists because you were awarded the Nobel Prize for Chemistry. I guess if you're doing crystallography, that's sort of fine. But in your case, I imagine it was a bit of a surprise to be awarded the chemistry prize.
It was indeed a big surprise. And in fact, I was telling somebody recently that I only succeeded in telling 2 of my 5 children personally about the prize because of Facebook. It spreads very rapidly. But one of my sons had learned about this almost immediately on Facebook. He lives in New York, and he immediately called his sister out in California, so it's 3 o'clock in the morning her time, all excited to say, Dad won the Nobel Prize. This was Wednesday morning. And she immediately said, no, he didn't. And he says, you're mistaken. He said, no, I just heard it. He won. She said, no. She said, you heard the prize was announced on Monday in medicine, and he didn't get it. He said, no, he got it in chemistry. I was like, chemistry? What's that all about? But, it is interesting. In my career, I've published hundreds and hundreds of papers. But the single publication vehicle in which I've published the overwhelming majority of my work is the Journal of Biological Chemistry. It's the single journal by far. There's not even a close second. So in that sense, I mean, if there's a term that could be applied to my work, I guess it's biochemistry or biological chemistry, which is a branch of chemistry. But yes, we've had some fun with the fact that we are now chemists.
I was going to go on and just ask last question. Does it matter how it's labeled? Does it matter whether it's labeled chemistry or biochemistry or pharmacology?
I think in a certain sense not, and I think what it shows is just how the interface between chemistry and biology and medicine, I mean, I've been called a lot of things in my time, not all pleasant, but I can handle chemist. I mean, over the years, people were always telling me, well, Bob, you'll win the Nobel Prize someday. And it was nice to hear, although it didn't seem to be happening. But if it happened, I always assumed it would be in medicine, not in chemistry. I think I have Brian to thank for that, no doubt about it.
Yeah, I think I might even take a little added delight in being awarded the chemistry prize. Of course, I think medicine and physiology is wonderful as well, but I've sort of been a chemist and wannabe for many years. Yeah.
Good company. Anyway, it was a great pleasure to speak to you both. Thank you very much indeed. And just remains for me to wish you a wonderful Nobel week in Stockholm.
Thank you.
Thank you.
Thank you.